Okay, everyone, welcome back to our next lecture video. This presentation will cover Chapter 25, which are the concepts of metabolism. Now, metabolism refers to all of the chemical reactions that occur in our bodies, and it's important that we understand metabolism before we start getting into the different types of tissues. So one thing that we can see on this page is this very confusing looking picture. I'll be the first to admit it. This picture is a frightening looking picture. Because this is metabolism. There's quite a bit to learn in metabolism. We do entire courses on metabolism. And so what metabolism, again, is referring to are all of the chemical reactions that take place within the body. So there are many reactions that sort of progress in certain directions, and some reactions can be reversed, and some other reactions can only go in one direction. So I sort of liken this. This sort of looks to me like a map of downtown Manhattan. So there's lots of different directions by which our reactions can proceed. So some of these, are going to be two-lane highways. Some of these are going to be a one-way highway, and we might even have some roundabouts in here. So this is the overall view of metabolism. Now, I'd like to assure you that we're not going to go and touch on everything here that we see in this picture. This is just to show you how intricate metabolism can be. But we're going to really focus on a special 4 reactions which make up a greater reaction known as cellular respiration. So as I just mentioned in the last slide, metabolism refers to all of the chemical reactions that take place in our body. And there are 2 types of reactions that are going to take place in our bodies. Those are catabolic reactions and anabolic reactions. Catabolic reactions are going to be decomposition reactions. They're going to break down substances into more simple parts. So we're going to take more complex molecules and break them down into smaller parts. Or we could say smaller, more simple molecules. Anabolic reactions are going to be the opposite. These are going to take simple molecules, and we're going to build up larger molecules with those simpler molecules. These are synthesis reactions because we're going to be forming a new type of molecule. And that new type of molecule is going to be more complex than the simple substituent parts. So thinking about our major macromolecules, proteins, lipids, carbohydrates, and nucleic acids, all of those macromolecules that we've learned up to this point, they're all going to be able to undergo some type of reactions, and that is through the dehydration, synthesis, and hydrolysis reactions. So one way that we can look at each of these reactions is that polysaccharides, large carbohydrates, are going to be broken down into monosaccharides. That's going to be a catabolic reaction. But if we were going to reverse this, and we could say that monosaccharides are going to be built up into polysaccharides, and that would be an anabolic reaction. So, just as a reminder from a few presentations ago, catabolism refers to the breakdown of molecules or crushes molecules, and anabolism refers to the assembly and the building up of more complex molecules from smaller molecules. So catabolic and anabolic reactions are always going to be coupled to ATP. ATP is our body's energy currency. So metabolism, referring to all of the different chemical reactions that occur in the body, can be separated into these 2 types, catabolic and anabolic. The catabolic reactions are going to break down larger molecules, and the anabolic reactions are going to build up those more complex molecules from smaller molecules. And ATP is the player that's going to couple these 2 reactions together. So one thing that we can see on this slide is that simple molecules, such as glucose, or amino acids, glycerol and fatty acids are going to come together to make lipids. Amino acids are going to be the substituent parts of our proteins. These are going to be our monomers of proteins. And glucose is going to be a monosaccharide. So these are all simpler molecules. And they're going to run through anabolic reactions. They're going to get built up and make complex molecules. So we can follow the arrows and see how these reactions are going to progress. So these more complex molecules are things like glycogen, proteins, and triglycerides. So these are the more complex versions of all of these smaller parts put together. Now through an anabolic reaction, we're going to require ATP, and you can see this arrow as it comes through here. This is indicating that ATP is going to be required in order for our anabolic reactions to progress. And through this heat is going to be released. We're going to be releasing energy in the form of heat from these reactions as well. But more importantly than heat, ATP is necessary here. ATP, our energy currency, if we can remember, has 3 phosphate groups. Well, when we break that single phosphate group off of the end, we're going to have 2 phosphate groups that are still attached to the initial adenosine molecule, but we'll also have a unique phosphate all on its own. So ATP, when it's hydrolyzed, it's going to form what is known as ADP and phosphate. This is adenosine diphosphate. So all we've done is take this molecule, ATP, and slice off this phosphate group here. And that leaves us with an ADP and a phosphate. Now these larger molecules are eventually going to get broken down. If we are to take these larger molecules into our bodies and eat them and start digesting those molecules in the form of proteins and in glycogen and such, we're going to start breaking those molecules down through catabolic reactions. Now catabolic reactions are going to transfer energy from the complex molecules to the ATP. So this is very interesting because they actually form ATP through the catabolic reactions. So you can see how ATP is coupling these two reactions together. While ATP is playing a major role in both of these, you can see ATP is going to be formed by catabolic reactions and then reused over here next to anabolic reactions. So this is a nice slide to show you how ATP is going to be coupled to both of these types of reactions, and this is an overall view of metabolism. The next concept we're going to touch on is energy transfer. We could also call this electron transfer. Electrons are going to be carrying energy with them. So think back to the redox reaction or the oxidation-reduction reaction. This reaction is going to function to transfer one electron from one particle to another. And so the oxidation-reduction reaction or the redox reaction is an energy transfer reaction because we're just transferring one electron from one molecule to another. One thing I want to remind you about oxidation-reduction reactions is that oxidation refers to a loss of an electron and reduction refers to the gain of an electron. So when one thing is oxidized, something else is going to be reduced. So we'll look at the oxidized substituent first. This reaction down here is showing us an oxidation. So this is lactic acid. This is the molecular structure of lactic acid. Now I don't need you to know the molecular structure of lactic acid, but I want you to get the concept from what we're going for here. So oxidation is the loss of an electron. So through an oxidation, lactic acid becomes pyruvic acid, which is just the molecular structure of pyruvic acid here. Now the real only difference that we see is that we've lost a hydrogen off this side and we've lost a hydrogen off this side. Now the way that these different chemicals are going to bond together, these hydrogens are going to take their electrons with them. And so this molecule is going to have less electrons than this one will. So this was an oxidation reaction. This molecule has lost electrons. Now whenever we undergo an oxidation, we decrease our potential energy because if we are losing electrons, we're losing energy. So I want you to imagine that whatever molecules have the most electrons are going to have the most energy. And that is shown for us here. The more electrons we have, the more energy is going to be found in our molecule. So this is an example of an oxidation reaction, and now we're going to look at a reduction reaction. So here is a reduction reaction. This is just the opposite reaction from what we saw on the last slide. So pyruvic acid is going to be reduced and become lactic acid. So these are just opposite reactions. We've just turned the arrow over and we're going in the other direction here. So again, we've taken reduction. Reduction is the gain of electrons. So pyruvic acid, when reduced, becomes lactic acid. We've added these electrons back on and we get these hydrogens back. And so we're left with lactic acid once again. So oxidation and reduction reactions are sort of opposites of one another. And this is the slide that we saw in our last presentation. So you can see that molecule A has the electron and molecule B does not. Molecule A is going to be oxidized because it's going to lose this electron to B through the reaction, and B is going to gain the electron, which means that molecule B is going to be reduced. And so the reduction is the gain of electrons. And if we were to turn this arrow on its head, everything else would be reversed here. So oxidation and reduction reactions can go in either direction. Now I need to mention that whenever we have some type of oxidation occur and we lose those electrons, those electrons aren't just disappearing. They're having to go somewhere. And they are transferred by these two coenzymes. So these coenzymes are nicotinamide adenine dinucleotide and flavin adenine dinucleotide. Now, what a mouthful that is. So we're going to call these NAD and FAD. That's our way we're going to refer to these. And so when these are carrying hydrogens and electrons, we're going to call them NADH and FADH2. If we don't have any hydrogens, we'll just call them NAD+ and FAD. So these two coenzymes are going to be so vitally important in cellular respiration. These are going to be the molecules that carry electrons to other places in our cells that we can use to produce energy. So these different coenzymes are going to be vital for taking these electrons somewhere else to form energy. So once again, we can just see here that this reaction, NAD plus is going to be the oxidized form, and when we add these electrons and the hydrogens to our NAD, that gives us NADH, and this is the reduced form. And the reduced form of FAD is going to be FADH2. And this reaction can go in the other direction and give us an oxidation instead. So we're going to see now how lactic acid is going to transfer its electrons to NAD plus. The reaction here, you can see the reactants are going to be lactic acid and NAD+, and after we undergo this reaction, we're going to get the products pyruvic acid and NADH with this hydrogen here. So lactic acid is the reduced form. You can go back to the last slide and see that lactic acid is the reduced form. It's going to have more electrons, which means it's going to have more potential energy. Now, NAD plus is the oxidized form. So as we react these two molecules, as these two molecules here meet in the middle, the electrons from lactic acid are going to transfer over to NADH. And that means that this is going to get oxidized while this is going to get reduced. So this is how we will see an oxidation-reduction reaction with two different molecules, allowing for this energy transfer. Oftentimes when oxidation reactions progress, they're going to release some energy, and that energy can be used to create ATP. So here we have adenosine diphosphate. This is ADP, adenosine, with two phosphates here, and we have this plus here showing us that phosphate is its own entity, as well as the energy from our oxidation reaction that's been released. And if we're able to take this energy, we can use it to form a bond between these two phosphates to give us ATP. This is adenosine triphosphate. This type of reaction is called a phosphorylation. Phosphorylation is going to increase ATP's overall energy, and we can do more things with this molecule. ATP is our energy currency, so this is a very important molecule that we have in our bodies. And the entire purpose of cellular respiration in chapter 25 is telling us how our bodies make ATP. That is the whole purpose of this chapter. So that will take us into carbohydrate metabolism. Carbohydrate metabolism will include the four major reactions that give us cellular respiration. Carbohydrates are going to be our saccharides, and glucose is going to be our most notable carbohydrate in this presentation. So when we eat carbohydrates, we're going to be taking in these different saccharides and breaking them down into their monomeric forms. So glucose, fructose, and galactose, we're going to have all of these different types of carbohydrates floating around after we have digested whatever food it is that we have eaten. So when we eat, we bring in carbohydrates, and that's going to break those carbohydrates down through digestion into these more monomeric forms. So while there are different types of carbohydrates and different monomers of carbohydrates, all of these carbohydrates are going to get converted to glucose during digestion. So carbohydrate metabolism really refers to glucose metabolism because glucose, everything gets converted to glucose. So these four reactions are going to be based on glucose. So glucose is such an important molecule. We use glucose for anything. We use glucose to create ATP, to synthesize glycogen and triglycerides. We're going to make amino acids with glucose. So glucose is going to be such an important molecule because it's going to help produce the energy that our bodies are going to use for everything. So to produce ATP, which is going to be the big topic of this presentation. To produce amino acids, we're going to need ATP to make new amino acids and glycogen and triglycerides and our macro molecules that we've learned in previous chapters. So glucose is going to be helping to make all of those larger molecules. So this here is the picture of carbohydrate metabolism. There are four different reactions that make up what is known as cellular respiration. Those four reactions are glycolysis, the formation of acetyl coenzyme A, the Krebs cycle, and the electron transport chain. These four reactions make up cellular respiration. Now, the purpose of cellular respiration is to take glucose and turn it into ATP. That's what cellular respiration is. We're going to start with glucose and we're going to turn it into a bunch of ATP that our body can use. And that's what we can see in this picture here. Glucose is going to be starting cellular respiration. Glucose is going to proceed through these 4 reactions. It's going to be changed and broken down, and eventually it's going to give us a bunch of ATP. So that is the main purpose of cellular respiration, to take glucose and turn it into ATP. So we are going to go over each of these 4 reactions. Each of these 4 reactions will have unique characteristics, will have unique reactants and unique products, unique energy outputs, and so forth. Most of those things can actually be found on this slide. So I would actually make a recommendation that you study this slide in this picture when you are looking over metabolism. So I'm going to go over each one of these, but I'll come back to this slide, and I'm going to show you that this picture is going to be very helpful and contain most of the information that we actually are going to need through chapter 25. So the first reaction in carbohydrate metabolism or cellular respiration is glycolysis. The purpose of glycolysis is to take glucose and turn it into 2 molecules of pyruvic acid. We briefly talked about pyruvic acid when we mentioned the oxidation and reduction reactions. So that's what glycolysis is. We're taking glucose and turning it into pyruvic acid. Pyruvic acid is then going to be turned into some other things, and then those things are going to be turned into some other things, which will eventually give us ATP. But in terms of glycolysis, all we're really looking at is glucose being turned into pyruvic acid. Glucose has 6 carbons, and we're going to slice this molecule in half and give us 2 molecules of pyruvic acid, which will each have 3 carbons. So we're basically just taking glucose and slicing it in half. Glycolysis is going to include 10 reactions and occur in the cytosol of our cells. And that is shown here on this picture right here. So this is the mitochondria. We can see that the mitochondria is going to be having our double membrane. This is going to be the outer membrane. And this is going to be the inner membrane with those cristae that we learned about in chapter 3. These are the cristae of the inner mitochondrial membrane. And out here is the cytosol. So in the cytosol, glycolysis will be turning glucose into 2 molecules of pyruvic acid. So there's a few other things that I'm going to tell you about glycolysis, but that's the main idea. So this right here is glycolysis. Now here's another scary picture. I'll admit this is another scary picture. Glycolysis has 10 different reactions, and so glucose is going to become glucose 6-phosphate, and then glucose 6-phosphate is going to become fructose 6-phosphate, and then they're going to progress and progress and progress until we get 2 molecules of pyruvic acid. And each one of these reactions has its own enzyme, and that produces a different molecule with a different structure, and there's a lot going on here. This is not why I put this slide here. I just want to be able to show you that glycolysis is made up of 10 reactions, and we take glucose and turn it into 2 molecules of pyruvic acid. That's what you need to know about this. You don't need to know any of these other enzymes or molecules in here. You just need to know that glycolysis turns glucose into 2 molecules of pyruvic acid. Now, glycolysis as a reaction is going to require an investment. And when I say an investment, I'm talking about ATP. In order for glucose to become 2 molecules of pyruvic acid, we need to invest 2 molecules of ATP. So you can see that this reaction requires ATP, and this reaction requires ATP. Now, that doesn't sound like it's a good idea. We don't want to be giving away our ATP because that's what we're trying to make here. We want to make ATP. Well, it's okay because after we have invested these 2 molecules of ATP, we're going to be able to finish this reaction, and out as well as 2 molecules of pyruvic acid, we're going to get 4 molecules of ATP. You can see that 2 molecules are formed from this reaction. 2 molecules of ATP are formed here, and 2 molecules of ATP are going to be formed here. But we get one more thing. We get 2 molecules of NADH as well. So we took that NAD, we took 2 of those, and we were able to form 2 molecules of NADH. And that's why I say that this is an investment. We've invested 2 ATP now so that we can get back 4 ATP. So through every molecule of glucose, we're going to have to invest 2 molecules of ATP, and that will give us 4 molecules of ATP. So we get a net gain of 2 molecules of ATP and 2 NADH molecules every time one glucose molecule runs through the glycolysis reaction. Again, I know that glycolysis might seem rather scary, but that's what I need you to know. I need you to know that glucose becomes 2 pyruvic acid. I need you to know that we need to invest 2 ATP, and we get out 4 ATP and 2 molecules of NADH. As we go through each of these reactions, I want you to focus on where the reaction is going to be occurring, how many reactions are going to be making up each of these larger reactions, and the energy outputs that are going to be coming from each reaction. So our energy outputs for glycolysis are going to be 4 molecules of ATP and 2 molecules of NADH. Most of the 4 reactions that make up cellular respiration are going to require oxygen, but glycolysis, in fact, is going to be the one reaction that does not require oxygen. So glycolysis does not need oxygen in order to run. However, the Krebs cycle and the electron transport chain will both require oxygen, and if we don't have oxygen present, we cannot progress through the Krebs cycle or the electron transport chain. Now, regardless of if we have oxygen present or not, glucose is going to become 2 molecules of pyruvic acid. However, what happens after this is going to depend on whether or not oxygen is present or not. So if oxygen is scarce, we will call these anaerobic conditions. If anaerobic conditions are present, pyruvic acid is going to be reduced, and that's going to give us a molecule of lactic acid. We can remember the reduction reaction we saw earlier. So if there's no oxygen present, pyruvic acid is going to become lactic acid. However, if oxygen is present, and we'll call these aerobic conditions, if oxygen is present and we have these aerobic conditions, this is going to allow our pyruvic acid to be converted to a molecule known as acetyl-coenzyme A. And this will bring us into our second major reaction that will make up cellular respiration. And that is going to be the formation of acetyl-CoA. So what this slide is showing us is that glucose is going to run through glycolysis to give us 2 molecules of pyruvic acid. We can also call pyruvic acid pyruvate. So if I refer to it as pyruvate, they're the same thing. Now, this is a molecule of pyruvic acid. So don't let this confuse you. This is the same picture of pyruvic acid I showed you on the other slide. So already, this stuff is something that we've already learned. We've already learned glycolysis. That's all we see here. But depending on if there are aerobic conditions or anaerobic conditions, that's going to determine which product is going to come after this. So if oxygen is present, which we want oxygen to be present so that we can create acetyl-coenzyme A, pyruvic acid is going to undergo an oxidation, and that will produce acetyl-coenzyme A, which is this molecule right here. But if oxygen conditions are low, that's going to cause a reduction and give us lactic acid. Lactic acid is not going to be able to continue through the rest of our chemical reactions. So we want to be able to get acetyl-coenzyme A in order to progress through cellular respiration. Now there are 2 other byproducts of the formation of acetyl-coenzyme A, and those byproducts are CO2 and NADH. So for every 1 molecule of pyruvic acid that comes out of glycolysis, that's going to give us 1 molecule of acetyl-coenzyme A, 1 molecule of NADH, and 1 molecule of CO2. CO2 is a waste product that we need to release from our body, and NADH is going to be another one of those coenzymes that's going to carry energy to our final step. So acetyl-CoA is what we want, and in order for us to get acetyl-CoA, we have to have oxygen present. We must have oxygen present in order to get acetyl-coenzyme A. Now, if we have lactic acid over here, and lactic acid is formed, we can add oxygen. If we add oxygen, that can take us back this way, and we can get acetyl-coenzyme A. But if we do not have oxygen present, lactic acid cannot enter the Krebs cycle, which is going to be our next reaction. So as we look at this, every molecule of pyruvate gives us 1 acetyl-CoA, 1 CO2, and 1 NADH. Now, keep in mind that 1 glucose molecule will give us 2 molecules of pyruvic acid. So if I was to ask the question on the test, 1 molecule of glucose is going to give us how many molecules of acetyl-coenzyme A? The correct answer would be 2. Because 2 pyruvic acids come from glucose, and each of those 2 is going to give us an additional acetyl-CoA and an additional NADH and CO2. If I was to ask you on the test how many molecules of acetyl-coenzyme A are formed from 1 pyruvate molecule, that's going to be 1. So it's important to be careful in your understanding that glucose gives us 2 molecules of pyruvic acid, and each molecule of pyruvic acid is going to give us 1 of these. So the reactants of the formation of acetyl-coenzyme A are pyruvate, and the products are going to be acetyl-coenzyme A. This is just an educational picture so that you can see pyruvate and acetyl coenzyme A afterwards. So here are the reactants. We have this group here that's going to be added on to our pyruvate molecule, and we have this red group that's going to actually be removed. And so we're going to take this CoA group and attach this onto our ketone, and that's going to make acetyl coenzyme A. But what happened here? We also had NADH. That's going to pick up those electrons in the hydrogen here, which is going to give us NADH, which will be used later to produce lots of ATP. This molecule right here is going to be CO2, so this is where CO2 comes from. So this is just an educational slide. I'm not going to put this slide on your test, but I just want to show you how we can track some of these substituents as we go through these reactions. So to summarize acetyl coenzyme A, for every 1 molecule of glucose, we're going to get 2 molecules of CO2, 2 molecules of NADH, and 2 molecules of acetyl coenzyme A. So these are our energy outputs and our waste outputs. The NADH and our acetyl coenzyme A are going to be used in later reactions, and CO2 is going to be a waste byproduct. So every time 1 molecule of glucose is broken down through glycolysis in the formation of acetyl coenzyme A, this is what's going to be coming from this reaction. And always keep in mind that Acetyl-CoA formation will require aerobic conditions, and that means that oxygen is going to be required to progress this reaction. So to summarize glycolysis and acetyl-CoA formation, glycolysis is going to refer to the conversion of glucose into 2 molecules of pyruvic acid. We can see this reaction shown here, and this occurs in the cytosol. Now, pyruvic acid is going to be transported into the inner mitochondrial membrane. We'll say that this is now going to be in the mitochondrial matrix. That's going to be the space inside of our inner mitochondrial membrane. And we got some energy products from glycolysis, but from the formation of acetyl coenzyme A, you can see that pyruvic acid is going to be converted into acetyl-CoA over here. And we're going to get our NADH and our CO2, and this reaction occurs inside of the mitochondria. So glycolysis occurs in the cytosol, while the formation of acetyl coenzyme A occurs in the mitochondrial matrix. Now there's one other reaction that occurs in the mitochondrial matrix, and that is the Krebs cycle. The Krebs cycle will also occur within the mitochondrial membrane. So inside of that inner mitochondrial membrane is where our Krebs cycle is going to be found. So once again, we can see glycolysis and the formation of acetyl coenzyme A that have been shown here. We can see that glycolysis is going to be giving us some ATP and some NADH. We can see that the formation of acetyl-CoA is going to give us some CO2 and some NADH. And then that's going to also give us acetyl coenzyme A, which will be allowing us to go into the Krebs cycle. So in order for us to enter the Krebs cycle, we must have oxygen, and we also must have acetyl coenzyme A. Remember, lactic acid cannot enter the Krebs cycle, only acetyl coenzyme A can, and that's why this molecule is important and why we have to have oxygen present in order to have these reactions carry out. So as long as oxygen is present and we have acetyl coenzyme A, we can enter the Krebs cycle. Now acetyl coenzyme A is going to enter the Krebs cycle here, and it's going to undergo a series of 8 reactions. The Krebs cycle is a series of 8 reactions, and we have more energy and waste outputs that come from the Krebs cycle. So you can see that as we go around the Krebs cycle here, and we go around these series of 8 reactions, we're going to produce 3 NADH, and we're going to produce 1 FADH and 1 ATP as well as 2 CO2. So you can see all of the different energy outputs. So we have 3 NADH molecules, 1 FADH, 1 ATP, and 2 CO2 molecules. Now this is based on 1 molecule of acetyl coenzyme A. Remember that glucose is going to produce 2 molecules of acetyl coenzyme A. So we have to multiply all of these energy and waste outputs by 2 when considering glucose being run through this cellular respiration. So we will say for every 1 molecule of glucose, we're going to get 2 molecules of ATP, 4 molecules of CO2, 6 molecules of NADH, and 2 molecules of FADH2. So the Krebs cycle has a couple names that I would like you to know. One of the other names that we can call the Krebs cycle is the citric acid cycle, because acetyl coenzyme A will be converted into citric acid right after it enters. And we can also call this the TCA cycle, which stands for the tricarboxylic acid cycle. So both of those are two other names that are just referring to the same set of reactions that our Krebs cycle is. Now, as I mentioned on the last slide, the Krebs cycle consists of 8 different chemical reactions. And the Krebs cycle reactions will occur in the matrix of the mitochondria. So this is our scary picture of the Krebs cycle. Again, there's 8 different reactions, and each reaction is going to have their own reactants and products with a different enzyme that's going to be catalyzing those reactions. And we have outputs and so forth. And once again, I don't need you to know this entire picture. I don't need you to know this. What I need you to know is that acetyl-coenzyme A is going to be required to enter the Krebs cycle, and running through those 8 reactions, we're going to produce these different energy outputs, which, as I just mentioned, are 2 ATP, 4 CO2, 6 NADH, and 2 FADH molecules for every 1 molecule of glucose. So coming from the formation of acetyl-coenzyme A, we have pyruvic acid, and every pyruvic acid molecule is going to produce a CO2, an NADH, and an acetyl-coenzyme A as long as oxygen is present. And as long as oxygen is present, acetyl-coenzyme A will be allowed to enter into the Krebs cycle and produce these different energy outputs. So we can see the energy outputs are listed for us here for the Krebs cycle. For every 1 molecule of glucose, which refers to 2 turns of the Krebs cycle. So remember that. Remember we have 2 different turns for every 1 molecule of glucose. We're going to get 6 NADH, 2 FADH, 2 ATP, and 4 CO2. So again, take this into account on the test. I might ask, how many molecules of NADH come from the Krebs cycle from 1 molecule of glucose? Or I very well might ask, how many molecules of NADH come from 1 molecule of pyruvic acid or 1 molecule of acetyl-coenzyme A? And if I ask for pyruvic acid or acetyl-coenzyme A, you're just going to take this number and cut it in half. So these are going to be the products from every 2 turns of the Krebs cycle. Now I want to take a quick second to come back to this slide to show you why I like it so much. So with each reaction, what we can see on this is the reaction itself that's been labeled, but each reaction is going to show us the reactants and the products. And then here for the formation of acetyl-CoA, we'll see the reactants and the products. And so each of these, this picture is nice because it shows us the reactants and products, but it will also show us the energy outputs. So from glycolysis, we get 2 molecules of ATP and 2 molecules of NADH. From the formation of acetyl-coenzyme A, we're going to get 2 molecules of CO2 and 2 molecules of NADH. From the Krebs cycle, we're going to get 2 molecules of ATP, 4 molecules of CO2, 6 molecules of NADH, and 2 molecules of FADH. So while there are other pieces of information that I want you to know, like where these reactions occur and how many reactions make up each of these, this slide is going to serve as a nice summary slide to show you those energy outputs and which reactants and products are important in the grand scheme of cellular respiration. Now, as we've gone through cellular respiration, we've constantly been creating this NADH and FADH. Now, these molecules are not going to be very useful over here. However, remember that these molecules are coenzymes that carry energy. So these NADH molecules and FADH molecules are going to bring that energy over to our 4th and final reaction of cellular respiration, which is the electron transport chain. While NADH and FADH are not a type of energy that our bodies can automatically utilize, they serve as a suitcase. They package up energy in a suitcase, and they're going to take this suitcase over here to the electron transport chain. So up until this point, we've only produced 4 molecules of ATP. We've only had a net gain of 4 molecules of ATP. Now, we're going to need a little bit more ATP than that. So the majority of ATP comes from the electron transport chain. Now, the electron transport chain is found in the inner mitochondrial membrane. So the location of this reaction will be in that inner mitochondrial membrane. So glycolysis occurs in the cytosol. The formation of acetyl-coenzyme A and the Krebs cycle will occur in the inner mitochondrial matrix, and then these NADH and FADH molecules are going to move their way over to the inner mitochondrial membrane where they can find the electron transport chain. The electron transport chain is a series of transmembrane carrier proteins that are found in our inner mitochondrial membrane. And these are going to be functioning as pumps. They are going to be pumping protons from the inner mitochondrial matrix out of the inner mitochondrial matrix. So I'll go back to that slide in just a second, but I just want to show you again that these series of carrier proteins are going to be found in the inner mitochondrial membrane. And this is the electron transport chain. And we have electron transport chains found all over the inner mitochondrial membrane of our mitochondria. This is why we have cristae, so that we can fit more of these electron transport chains within our mitochondria. So each of these proteins that are found in the membrane are going to be pumping protons. They are proton pumps, and each of them has their own name, but the only one that I want you to focus on is this last one here, which is ATP synthase. Now, again, I don't need you to learn all the mechanics of the electrons transport chain, but I do want you to understand why NADH and FADH are going to be so important. So what we can see in this picture is that NADH and FADH that has come from all of these other reactions is going to float over here to the inner mitochondrial membrane, and that's where we're going to start oxidizing these different chemicals, which will eventually allow us to form ATP. So the way that these are going to work is that the NADH molecules are going to give their electrons to these different pumps. And so the electrons are then going to energize these, and that's going to allow them to pump protons. So what happens is when we're pumping protons, which are just hydrogen ions, when we're pumping protons into the space between our outer and inner mitochondrial membranes, this is going to set up a gradient. We're actually going to be pumping more hydrogens out of the inner mitochondrial matrix, and this is going to create a higher concentration of hydrogen pumps over here. So we've now set up a gradient that's going to point back towards the inner mitochondrial matrix. So what do we know about gradients? Things will flow from high concentration to low concentration. So those protons are going to come over here to ATP synthase, and they're going to run through this transporter. ATP synthase is an enzyme that synthesizes ATP, and it functions as a crank. It's going to have ADP and phosphate, and when hydrogens are going to be coming through this enzyme, that's going to sort of force the ADP and the phosphate together to produce ATP. So that is why ATP synthase is the enzyme that I'd like you to know. I don't need you to know all the other names for these other ones, but I would like you to know that ATP synthase is the major enzyme that's going to produce the most ATP within cellular respiration. So let's think back to NADH and FADH. They were taking electrons to these different pumps found in the inner mitochondrial membrane. That's going to energize the pumps, which will allow the protons to get moved to the inner space between the outer and inner mitochondrial membranes, which will allow us to crank this ATP synthase to form ATP. So truly, the majority of ATP that comes from cellular respiration is actually coming from NADH and FADH as well. So again, when our carrier proteins are reduced, that's going to energize them, and it allows them to pump protons into the space between the outer and inner mitochondrial membranes. So overall, the electron transport chain is going to offer us 26 to 28 ATP. So this is a lot of ATP compared to all of the other reactions in cellular respiration. So NADH and FADH are going to be facilitating the formation of this ATP. Now it's 26 to 28 because depending on the type of cell, certain things can be a little bit different. NADH and FADH2 actually will offer us different amounts of proton pumpage. So keep in mind that this does have a range. So on the test, if I was to ask how much ATP comes from cellular respiration as a whole, we're going to take the 4 ATP that we got from our other 3 reactions, and we're going to take this 26 to 28 to give us a grand total of 30 to 32 ATP. And just like all of our reactions except for glycolysis, the electron transport chain will also require oxygen. Oxygen is going to be used here in the electron transport chain to combine with these electrons, and that's going to help us to form water. So this is an understatement. The electron transport chain is the major source of ATP generation in our bodies. We get 30 to 32 ATP from the entirety of cellular respiration, and 26 to 28 of those ATP molecules are coming from the electron transport chain. So for every 1 molecule of glucose, we get 30 to 32 ATP. So for cellular respiration as a summary, I hope this slide is not overly confusing for you all. Glycolysis is going to produce 2 ATP and 2 NADH. Those are going to be the net gain. And by net gain, I mean the overall gain. We had to invest 2 ATP, so we had to pay ourselves back for that 2 ATP. So while we actually produced 4 ATP in total, we just paid ourselves back. And so our net gain is 2 ATP and 2 NADH from glycolysis. The formation of acetyl coenzyme A is going to give us 2 molecules of NADH, and there's going to be some carbon dioxide as well, but these are just referring to the energy outputs. The Krebs cycle is going to give us 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of ATP. So if we add up all of the energy outputs from glycolysis, the formation of acetyl coenzyme A, and the Krebs cycle, we'll get 4 ATP, 10 NADH, and 2 FADH. The NADH and the FADH are going to travel over to the electron transport chain, and that's going to allow the electron transport chain to generate 26 to 28 more ATP than we had, for a grand total of 30 to 32 ATP for every 1 molecule of glucose that is run through cellular respiration. So we're back to my favorite slide. I definitely recommend that you study this slide. So what we can see is that we have glycolysis, the formation of acetyl coenzyme A, the Krebs cycle, and the electron transport chain. Those are our 4 reactions that make up cellular respiration. The reactant of glycolysis is going to be glucose, and the product is going to be 2 molecules of pyruvic acid. Now, the formation of acetyl-CoA is going to have its reactants be these 2 molecules of pyruvic acid, and they are each going to produce 1 molecule of acetyl coenzyme A for a grand total of 2 molecules of acetyl coenzyme A. The acetyl coenzyme A is going to be allowed to go into the Krebs cycle as long as oxygen is present. And on the sides here, we can see the energy and waste outputs that come from each of these reactions. So glycolysis is going to give us a net gain of 2 molecules of ATP and 2 molecules of NADH for 1 molecule of glucose. The formation of acetyl-CoA is going to give us 2 molecules of CO2, 2 molecules of NADH from 1 molecule of glucose. And the Krebs cycle is going to give us 2 molecules of ATP, 4 molecules of CO2, 6 molecules of NADH, and 2 molecules of NADH per 1 glucose. So all of these energy outputs are calculated based on 1 glucose. So if you learn these energy outputs, and I was to ask you about how much comes from pyruvic acid or how much comes from acetyl coenzyme A, you can just cut this number in half, and that will give you the correct answer for how many energy outputs or waste byproducts come from each of these molecules. And then over here at the electron transport chain, you can see that oxygen is also going to be required, and that's going to help us to form water, and we will get a total of 26 to 28 ATP. It's going to be the NADH and the FADH that are going to be feeding into the electron transport chain. So you can see that these lines are directly correlated with NADH and FADH alone. Those are going to be what come over to the electron transport chain, energize the electron transport chain, and allow it to produce the majority of ATP for the human body. Now, as important and vital to our body's function as cellular respiration is, there are other metabolic reactions that take place within the body. So when we eat a meal that's very high in carbohydrates and we have glucose, and all of our other monosaccharides have been converted to glucose, and we bring those and absorb them into our bodies, some of that glucose is going to be used to form ATP. But we might not necessarily need all of the ATP from all of the glucose that we had in our meal. So some glucose can be stored, and that is going to be stored in, you guessed it, glycogen, our primary storage form of polysaccharides in the body. So remember, glycogen is just a long chain polysaccharide that's going to be made up of different glucose monomers. It's just glucose that has been linked together, and that is going to be our storage form of glucose that's found in the body. Glycogen is going to serve as a glucose reservoir, so when energy is needed, we're going to be able to access that glycogen, break some of those glucose molecules off, and use those glucose to make ATP. So each of these processes is given its own reaction. So on the left here you can see glucose, and on the right here you can see glycogen. So when we take glucose and we store it as glycogen, that's going to be referring to glycogenesis. Genesis is the formation of glycogen. So glycogenesis is going to be forming glycogen. Now when we need to break down glycogen to form glucose, that's going to give us glycogenolysis or glycogenolysis. So glycogenolysis or glycogenolysis is referring to glycogen being turned into glucose. So this here will be an anabolic reaction because we're taking simpler molecules and turning them into more complex molecules. And glycogenolysis is going to be a catabolic reaction because we're breaking down this more complex molecule and we're going to be producing glucose. And that is going to eventually enter into cellular respiration so that we can break that glucose down and make more ATP. So gluconeogenesis is another reaction that we have within the realm of metabolism. It's very possible that our glycogen stores might run out. This can occur if we are starving or fasting. So if our bodies are in need of more glucose, we can actually synthesize it through this reaction, gluconeogenesis. Now, gluconeogenesis is just the reverse of glycolysis. We take all of these other reactions, we turn them in the other direction, and we take pyruvic acid to make glucose. So this is helpful if we need to form glucose for some reason. Now, if we are out of pyruvic acid, there are other ways for us to make this molecule as well. We can take lactic acid, and we can use that to give us pyruvic acid. Remember, lactic acid was a byproduct of acetyl-coenzyme A formation that did not have oxygen present. So we can take that lactic acid, and we can turn the reaction around, and that can give us pyruvic acid to go in this backwards direction. We can also have a glycerol that will come from fatty acids. Remember, our lipid structure are different fatty acids and a glycerol backbone. So the glycerol molecule can feed into this reaction as well to help us form glucose also. Certain amino acids can also feed in here. So there's lots of different types of molecules that can feed in here and help to produce glucose through the reaction gluconeogenesis. So I realize at this point that I've just thrown a bunch of reactions that all start with G, and they all sort of sound the same. So that is the purpose of this slide. So what I want to show you here is that glycolysis, gluconeogenesis, glycogenolysis, and glycogenesis are all going to be different reactions. They all are spelled slightly differently, and they all refer to something different. So glycolysis was our first reaction in cellular respiration. This refers to the catabolism of glucose. We are breaking down glucose with glycolysis, and we're forming pyruvic acid. Gluconeogenesis is the opposite of glycolysis. Gluconeogenesis is the anabolism of glucose. We are forming glucose. We're building up glucose from other different molecules. So these reactions will be reverse reactions. They will go in opposite directions as one another. Glycogenolysis, on the other hand, is going to refer to the catabolism of glycogen. We are breaking down glycogen. So it's possible that we might need to liberate glucose from glycogen, and we can use that to create more ATP, and that's going to be done through glycogenolysis. Glycogenesis is our fourth reaction here. This is the anabolism of glycogen. So we are forming glycogen here. So I'm going to give you some tips and tricks to remember these different reactions. So first and foremost, any type of anabolic reaction will end in genesis. Anabolism refers to the formation of more complex molecules. So we're going to be using this suffix here, genesis. So any word that ends in genesis is referring to an anabolism. So you can see that these are the two reactions that are anabolic. Lysis, as I refer to so frequently, is going to tell us which reactions are catabolic. So lysis reactions are going to be catabolic because we're taking large molecules and we're breaking them down. So the suffix here is going to tell you which reaction we're looking at here. If the ending is genesis, that's an anabolic reaction. If the ending is lysis, that's a catabolic reaction. Now, in terms of glucose versus glycogen, how do we tell the difference? Well, if you look over here, if you see glycogen in the reaction's name, it's going to be referring to glycogen. So glycogenesis, glycogen, you can see glycogen written here. That is referring to the anabolism of glycogen. You can see that glycogen's also written here. So that's how you'll be able to distinguish whether or not these are anabolic or catabolic reactions and whether or not they're referring to glucose or glycogen. So we're going to briefly touch on lipid metabolism. Lipids are very nonpolar, which means that they are hydrophobic and do not combine well with water. These are things like our olive oil. Olive oil does not mix well with water. Oil and water do not go together. They are hydrophobic. And so in order for them to get transported throughout the body to different places, we have to have our lipids combined with some type of proteins. Those are called lipoproteins. The reason that we have to have lipoproteins transport lipids throughout the blood is because blood is going to be mostly water. So blood and lipids are not going to combine well. So we have to have lipoproteins in order to transport lipids across our bodies. So when we look at lipid metabolism, you can see that we have two major reactions here. We have lipolysis and we have lipogenesis. Now, applying the same understanding that we did in the last few slides, lipogenesis is going to be lipid anabolism, and lipolysis is going to be lipid catabolism. So it's important to know that we can actually use lipids if we need to make more glucose or more ATP. Lipids, in the form of triglycerides, can be broken down and help us to form more ATP or more glucose if we need to. So triglycerides are, the structure of triglycerides is a glycerol backbone and 3 fatty acids. You can see that glycerol can come in this direction. Glycerol can move here and feed into glycolysis. And then it can come into and form acetyl-coenzyme A, and the rest is history. Fatty acids can also come in here and form acetyl-coenzyme A as well. So triglycerides can also be used to create ATP if we do not have glucose present. If we're out of our glycogen stores and there's no glucose present, we can use triglycerides as well. We can also use certain amino acids. Certain amino acids can feed into form acetyl-coenzyme A, and they can also feed into the Krebs cycle at other places. The green arrows here show lipolysis reactions, and the orange arrows show lipogenesis reactions. So lipogenesis, referring to the formation of lipids, are going to move in this direction. So lipogenesis will form triglycerides. Lipolysis will break down lipids. Now, if we take in more triglycerides than our body is going to need, they're going to be stored in adipose tissue, and adipose tissues are our fat cells. So triglycerides are a storage form of fat that we will find in our adipocytes. So what I want you to understand from this picture is that if we don't have glucose, we can use other things to make ATP as well. We can use triglycerides and fats. We can use certain amino acids, and those things can feed in to form acetyl-coenzyme A, and that will allow us to progress to produce more ATP. Now, one thing I would like to mention is that acetyl-coenzyme A, a lot of these reactions here go both ways, but the acetyl-coenzyme A cannot be converted back into pyruvic acid. So we cannot take fatty acids and turn them into glucose. That's something I'd like to mention here. Acetyl-CoA cannot be converted back into pyruvic acid. Real quickly on protein metabolism, proteins can be broken down into amino acids, and those can also feed into the Krebs cycle as well. So you can see that the different names here, phenylalanine, tyrosine, leucine, lysine, these are all different amino acids, all highlighted here in yellow. All of these amino acids can feed into different places of the Krebs cycle, and that can also allow us to produce more ATP. Protein catabolism refers to the breakdown of proteins into their monomers, which are amino acids, while protein anabolism refers to the synthesis of new proteins from amino acids. So while we can use triglycerides to form glucose and ATP, we can also use amino acids to help us form ATP as well. So these are some summary slides. I suggest you give these a read. These will characterize the different reactions that we've talked about in this presentation. So make sure that you are feeling comfortable with these metabolic reactions. Lastly, for metabolism, during states of fasting or starvation, which are defined here, fasting is going to be without food for several hours. Starvation is going to be going without food for weeks to months. So these are very different states. But when our bodies are in a state of fasting or starvation, we start to produce another chemical known as ketone bodies. And ketone bodies can be used as energy for our cells as well. So that will conclude Chapter 25, Metabolism. If there are any questions, as always, send me a message or give me a call, and I will see you guys in the next video.